the Rosehip neuron, a new type of brain cell. Researchers in Hungary and America had been independently studying the human brain when they both discovered a new type of brain cell. The Hungarian researchers had been recording the electrical activity of the brain's inhibitory neurons, special cells that regulate the speed of electrical signals in the brain, ensuring a healthy balance of electrical activity. Over time, they began noticing cells with a distinctive shape that must have been formed by the densely packed network of nerve fibres around the cell centre. The shape reminded them of a rose after its petals have dropped, so they named these cells Rosehip neurons. American scientists also detected these cells using a new technique that allowed them to identify active genes in brain cells. The scientists could have continued their research independently, but they decided to collaborate in order to better understand these unique cells. The Hungarian team examined the shape and electrical features of Rosehip neurons, while the American scientists looked at their genetic characteristics. While the precise function of Rosehip neurons is still unclear, this research collaboration has provided the first steps towards a deeper understanding of their role. Since Rosehip neurons are one type of inhibitory neuron, they must be involved in slowing down the speed of the electrical signals from neighbouring cells. Additionally, Rosehip neurons comprised only about 10 to 15% of the inhibitory neurons studied and connected to only certain brain cells, leading researchers to conclude that their inhibitory function is very targeted. Scientists believe Rosehip neurons have the potential to provide answers to questions that have long troubled the medical and scientific community. For instance, Rosehip neurons may well be unique to humans. If so, their presence might be one of the factors underlying the superior cognitive abilities of humans.